we basically found now that susceptibility, although as a general rule, West Indians are more susceptible, there's a lot of very susceptible Guatemalans, a lot of very susceptible Mexicans, and very susceptible hybrids. So we basically want to get a handle on if it's only related to the rate of sap flow and the rate of movement of the fungus in the tree, or is there other things going on there? And so we have clonally produced trees of Mexican, Guatemalan, and West Indian races, which will each be inoculated with a fluorescent strain of the fungus. We'll also look at transpiration, sap flow, and like we're doing with the other study with in-tree movement of the pathogen. And xylem anatomy, the size of the vessels, the density of the vessels, how much tylosis there is, and the rate of infection will be determined. And so we're going to determine if anatomical and physiological differences among trees are, affected with in-tree movement of the pathogen in the Laurel Wilt, or is it due to other things as well? And the rationale for that is if we can, definitively relate avocado race to Laurel Wilt susceptibility, it will inform us at least about continued selection and breeding for Laurel Wilt tolerance, where we can hone in on. And as I said, these objectives were scheduled just to start last month, so we haven't really gotten very far. What we have done is hired a postdoc, Daniel Palberg, to work on these objectives. He started a few weeks ago. He's in the back there. And the other thing, Daniel has a really strong background, has a degree in chemistry, about 10 years in the chemistry lab. He also has a degree with working on chemicals that come from fungi, and the relationship with host trees. So in collaboration with Romina Gaziz's group, he's also going to be extracting chemical compounds from Loricola and antagonistic fungi to test their potential for disease suppression. So we're going to he's going to look at extracts, and he's going to see how they work, and if any of them are effective, then we're going to have to find out, what exactly is in them. And we've already obtained plant material to work with, so we're just getting started. And we also, it takes a long time to count xylem vessels once we harvest the trees. So while we were waiting to get started, we developed, with the help of a fellow, Sam, who used to be here, who's now a grad student in Germany, he helped us develop AI algorithms to quickly count xylem vessels and the number and density of xylem. And so that's what we've done so far, which isn't much because we just got started. Thank you.
Do I need to be introduced?
Yeah.
So thank you all for coming today, and thank you for the attendance online. My name is Malik Hamami, and I'm in charge of the economic assessment of the Laurel Wilt management strategies. So I'm among the team formed with Dr. Evans, Gilly, and Betty Bannon. Initially, the project was submitted by Dr. Evans, and I'm taking over since last year, replacing him on this project. So today I'm going to explain why the economic assessment is important, because we have several questions on why are you guys always trying to keep track of the cost and revenues, and then is it profitable or not? The main question, and later I'm going to state the objectives and methods that we are going to propose, that we are working on right now, and then a progress update. So why is it important? Why all these economists at any project walking around with a survey, and they are always asking about the cost and how much it cost you, and whether it's profitable or not. So our colleagues in this project, they are working on scientific techniques and measures to combat Laurel Wilt, and they are effective, and they are showing how is it effective. But our role is to show that it's also economically efficient, and to efficiency leads to profitability. And I know that there is some work being done before, especially surveys have been distributed before, but it's dynamic. We know that production yield changes with time, input inflation is changing the input cost. And there is also, from the other side, an evolving demand. So there is a change in behavior and preferences. And for that, it's always needed to estimate what's the willingness to pay of the consumers, and that changed the whole dynamic of the market without forgetting the trade impact, right? So our role is to measure this profitability that was measured 10 years ago, I think in the previous phase, and to see if it's profitable or not. What was profitable 10 years ago might not be profitable now, and vice versa. Right? So also there is also climate insurance and policy considerations that are playing a role here. So to be sustainable, you need to be profitable. Of course, that's a common sense. So our objective, our economics objectives for measuring the economic assessment are to update farm production costs and evaluate the cost effectiveness of the new disease control treatments. We intend also to develop an interactive tool to visualize profitability changes from adopting proposed treatments, and explore barriers for new technology adoption. What constitute a barrier? How can we help you to adopt this better, or to convince you that it's efficient and effective? And that's all done through profitability assessment and their proposed treatments, accounting for risk and fluctuating prices and yields conditions. So for that, we will need to do a status quo, how much it costs now to grow an acre of avocado here in this area, and then we are going to simulate those yield changes due to adopting those measurements. And given that yield change and also the market, the external conditions of the market changes, we are going to see whether it's going to be profitable or not. And finally, we are going to update, do frequent updates on the changes in Florida avocado crop insurance program. So the progress update is that we have obtained the IRB approval, and we have the surveys ready. We already distributed several surveys. Several people here have already answered. So we have interviewed 4 growers so far that accepted to provide the feedback, and that's 25% of the industry. We have recruited 2 graduate students that are partially on this grant, but on other grants as well, doing economic assessment of the avocado Laurel Wilt related issues, broader issues, like we can even include trade in there. And we have drafted scientific article on avocado, Florida avocado demand, supply, and the impact of Laurel Wilt, which are underway, that are going to be published ultimately. So the next steps is to complete the sample. We have 4 growers so far, and after discussions with Dr. Crane and the several growers, we know that there are several growers up north, mid Florida, that also are growing avocado, and that's different conditions. So that's different survey, and that's a different profitability. So we're trying to be proactive and try to have a sense of what's happening up north too, and collaborate with the agronomic team. So we need to get the feedback from their results on how is the new measures, how are they yield improving or cost reducing, so that we can simulate our profitability analysis. And then lastly, conduct the cost benefit analysis, mainly to compare profitability of current versus proposed disease management strategies, and incorporate the risk scenarios of yields and prices. And then finally, of course, like to publish avocado or Laurel Wilt related work, and to have an output out. Thank you.
Any questions now?
You're gonna expand on.
Okay. Yeah, well, thanks for the opportunity to present today. So so I'll be presenting on behalf of our team. So you've heard from Romaric Mwafo-Chinda before, who's working as a postdoc. And right now we're looking for another postdoc to continue the work. And Erin Plexula, a PhD student, is helping with our progress in the meantime while we're looking for the new postdoc. So we're looking at larger scale processes and how to manage at regional and national levels. So our main questions are how epidemics are likely to expand geographically, what recommendations we can give for surveillance strategies by anticipating where invasion is likely to happen next, and then input for regional management policies and regulations, how successful they're likely to be in the kind of simulation studies we can do to evaluate, based on the assumptions of this scenario, how well would a particular policy work? So some highlights of previous research. So we've developed a model where we can look at evaluation of policy effects. And Berea Etherton was a previous student who had led those efforts. We've looked at how host density dependence and environmental factors affect Laurel Wilt, Robin Chaudhry, working on that in the previous USDA project. So we've been developing models and code to make it easier to evaluate geographic connectivity. And I'll say a little bit more about that in a moment. And also Romaric Mwafo-Chinda's work to look at geographic risk, which I'll also say something more about. Oh, is there a question?
No, sorry.
Oh, okay. Okay, thanks. Yeah, and so the work of Berea Etherton and our team there, so there we were looking at some of the components of regional management and how policy can influence them. So for example, how good is the quality of management available? So that'll influence decision-making. And then what are the other factors and potential incentives that can influence management in a region? So that study gets into the details of that. And then so that was the past study of Berea. And so then in coming up with, when we hire the new postdoc, we'll look at more policy implication studies, building on the models developed before. So one of the projects of our lab is to build this plant health toolbox in general that we can use for larger scale analyses. And so that's had a lot of contributors over time. And we're incorporating these other sorts of larger scale processes in it, including a new app for to support implementation of expert knowledge elicitation. So asking experts for their opinions in a structured way to understand systems. And then mentioned this GeoHabNet R package. So that's designed for looking at habitat connectivity, or in our case, for Laurel Wilt, habitat can be defined to some extent, at least, in terms of the availability of the hosts and environmental conditions that are suitable for vectors and pathogen. So in this cropland or habitat connectivity analysis, we can look at, for example, are there big breaks in host availability which would slow down spread? Or are there important locations that are a bridge between different geographic regions that make it easier for spread? So then we can understand the importance of different locations that might be bridges or hubs in the potential spread of the pathogen. And so thinking about the spread and in the United States, and interesting to track it as it keeps progressing. So then the work of Romaric Mwafo-Chinda and Erin Plexula and our other members of the team in the USDA project and beyond. So here looking at climate similarity and host availability as two of the key factors. So one of the complications for evaluating climate conduciveness is that we know where the pathogen is, but we don't necessarily know where it could be, but has been limited by dispersal so far. So here we're taking a more conservative approach and saying, where is the climate similar, not necessarily limited by conduciveness, but where is the climate similar to where the pathogen has already been, where the disease has already been? So this is a preliminary map. We're still working to refine this some, but this is looking at two of those key factors: climate similarity to where the disease already occurs, and then host connectivity. So how much of the host is available, and how well connected is the landscape of the host, so that it makes it easier for the vector to move with the pathogen. So then what we're adding in new analyses. So scenario analysis for introduction in new parts of the US and surveillance priorities. So this initial analysis of Romeric Mafo Chinda, looking at climate and host availability. And then using this new app, GeoHabNet, we can look at simulations of spread across the region and figure out which sorts of invasion patterns are most likely based on what we know now about the pathogen and vector. And so also, like I mentioned, building some on these models of epidemics and how policy and regulations may influence them. So we've got that initial analysis by Maria Etherton and that team. So we're looking at a couple policy scenarios in Florida, and now we'll expand that to look at a wider range of potential policies and in other parts of the United States. So thanks for the opportunity to discuss this, and look forward to any questions you might have, or if you have, as you know, when we develop these scenario analyses so that we can ask what if questions. So if you have particularly important questions that I didn't mention that you think would be valuable for us to look at in these simulations, please let me know.
Anybody have any questions for Karen?
Thank you.
Thank you.
Yeah, thank you.
Ladies. Good morning, everyone. My name is Daniel Carrillo. I am the tropical fruit entomologist, and I'm coordinating all the entomology work of this grant. I'm working closely with a lab in California, UC Riverside, led by Paul Rockman Jones, and in Texas by Mamadou Setamo. Just gonna start with a couple of slides here, some background information that is important to have clear. So this is an insect vector disease. No, there's an insect that transmits the pathogen from tree to tree, and we discovered some time ago that the disease has different dynamics in native ecosystems than in avocado. In natural ecosystems, you have a vector that is very efficient. The red bee ambrosia beetle, or Xyloborus glabratus, is native to Taiwan. It's an invasive species, and it has killed millions and millions of native trees that are closely related to avocado. However, we learned a long time ago, this is actually a 2012 paper, that avocado is a poor host for this beetle, at least the Florida avocados. In native ecosystems, it's clearly a dominant species. We have very low populations. In all these years working with avocado and Laurel Wilt, we have collected a handful of beetles. The other ones, we have collected millions in natural ecosystems. We also discovered that several of our native beetles can transmit the pathogen, but they do it less efficiently than the red bee ambrosia beetle, significantly less efficient. Why? Because they carry variable loads of the pathogen, 50 to 30% relative to the red bee ambrosia beetle. They have lower transmission rates, variable associations with the pathogen. These beetles are also generalists. They attack other trees where the pathogen doesn't develop. And they have different tree colonization patterns. Some of the beetles that we have here are secondary species that only attack trees that are stressed by other factors. So our role in this project, we participate in two objectives. The first objective is to build preparedness in Texas and California by identifying the ambrosia beetles that they have in Texas and California, identify their fungal symbionts. So these beetles depend on fungi to develop. And so we're working with a pathology lab for that. And predict continental scale disease movement. This is related to our previous speaker. So we made significant progress in this first objective. In Texas and California, we are conducting surveys, sampling avocado groves in multiple ways with traps, collecting logs to identify the ambrosia beetle complexes in both systems. In California, we started really strong this year. There's a photo of Paul Brockman Jones. He's leading the survey in California. We have already 14 sites covering the main production areas, the 5 main production areas in California. And so far, we have collected beetles in 10 genera. We are conducting the identifications. And so far, it seems to be a completely different complex than the one we have in Florida. We only share 1 or 2 species. And next, we're going to try to collect some live beetles to send to Romina for fungal isolations. I think everything is in place for that. And we are going to be 2 years in California doing this. And in Texas, we just started send traps, lures. So hopefully next year, we're gonna be presenting similar information for Texas.
And there currently is no Laurel Wilt in either of those states?
There's Laurel Wilt in Texas, but not in avocado. Natural areas only, not in California. The second objective that has an entomology component is to determine how canopy management strategies affect ambrosia beetle flight activity in avocado orchards. And this stems from previous knowledge also. We know that all these secondary vectors are generalist forest insects. They like forest environment. They like all their groves with crowded canopies. They like shaded areas. And we have several studies on the flight behavior of these beetles conducted by Octavio Menocal, that is here in the back. Now he is helping us as a postdoc in this objective. So we've been trying to establish a management strategy that is basically make the crop environment less suitable for these beetles. They like this forest environment. The forest environments try to make it less like a forest. And specifically in this grant, we want to test how the pruning practices that the growers use here in Florida affect the flight activity of these beetles. So some growers do selective pruning, is removing some limbs of the tree periodically, hedging with a machine, topping or skirting, that is removing the lower part of the canopy. How do these management strategies affect the ambrosia beetle activity? This year, we started with a trial in El Limonar. It's a grove here south of us that many of you may know. Frank actually helped us with this experiment. We took a plot and we had alternated blocks of 3 rows of trees that were either hedged or not. And in the middle row, we placed some sticky traps on the trees to evaluate beetle landing on those trees, and we also have traps all over the grove to monitor ambrosia beetle activity. So the idea is to see if this hedging has any influence on the abundance of the beetles. And so far, to our surprise, we have only captured 38 ambrosia beetles in the sticky traps and 13 ambrosia beetles. I'm talking about the ones that we know are capable of transmitting the pathogen on the funnel traps. This is significantly low. We could capture this years back in this grove in one day. We will capture beetles by the buckets from this grove. So the beetle populations are significantly down. And I think most of you have noticed that in terms of management of the
How about the
disease as
surrounding
well.
area? The surrounding area, how has that changed? Or is it similar?
Well, this is a very large grove. And this is an old photo. And if you remember, this is where we had one of the Laurel Wilt summits, I think back in 2016 or something. And this used to be like a site of a nuclear bomb. But it was like a epicenter of the disease.
But
Nowadays,
around
it has
Properties around it, how has that changed?
in general,
Similar.
I think throughout the growing area here in Homestead,
Daniel,
you have seen a regrowth.
there was a large avocado due east of that grove that was abandoned for a number of years. It's gone now. So that.
So is it like raised, it's open air? I'm just
It-
trying to.
It's still
You know,
still
that's
ag
the
bed,
prime
but
area,
it's
but what
not.
about around the neighborhood of that area? Is
In
that
general,
also-
I see a reduction in the ambrosia beetle populations, and also, like Mark was saying at the beginning, regrowth in many groves. I think is regional, not only about this growth. And in this one in particular, you see that it's very uneven because it had was affected by the disease, and the trees are recovering. So
It's all way more open.
yeah, but all the trees are coming back. And these numbers are like for us, it's unthinkable that we have these numbers now. If you remember back then, we were talking about thousands and thousands of beetles weekly.
I guess my policies work.
Huh?
I guess my policies work.
So that eastern.
Yeah, it's gone.
It's gone.
Okay, so
For
this
7 years.
sorry, this is a challenging experimental. It's challenging to demonstrate this thing of the flight behavior of the beetles, and it's also we were lucky that we were allowed to do this in this particular grove, and they were pruning and at that time, and they allowed us to do this sort of experimental layout. It's not the ideal one. But now we're gonna move to Trek here and do experiments. Next year, we're gonna be doing experiments here at Trek where we can manipulate things a little bit better. The other objective with an entomology component is that together with this reduction in the beetle populations, we have also seen an increase in other organisms that are colonizing the beetle nests inside the tree, the complexity of those breeding sites. And so we have found a bunch of nematodes, mites, and other microorganisms that are colonizing the beetle breeding sites. So we're trying to explore that diversity and see if they can interfere with the beetle's ability to cultivate fungi and actually control the beetles. Very difficult research because nobody has really looked into this. So far we are concentrated in the mite species. I had a student, Marielle Berto, that has been working on this, going to different groves and collecting logs and opening them up and looking at the nests and extracting mites from them. Very laborious work. And so far, this year, she identified 10 species of mites already. Out of those 10 species, 4 are new species to science. We already described one. These Proctolelaps and Dendrolelaps are 2 new species. We're describing this one. Very complicated to work with. We have Aline Tassin, the Baxis and acarologist, is helping us with the characterization of these mite species. And we've actually done some work with this species, Saponivorous mite, Histiogaster arborsignis, that we see very often inside the beetle galleries. And we did some studies first to establish methods to rear them, to be able to study them. So we published a paper on methods to rear the mites, and we now have lab colonies to work with them. And also this other manuscript on interactions of this mite with the fungal partners of the beetles. So we wanted to know if they actually feed on Lauricola and other symbionts of these beetles. And so we recently published this paper, that the first product for this grant. And we will continue working on this system and evaluating the potential use of these mites in biological control programs for ambrosia beetles, but could be extended to other wood-boring insects. Yeah, and that's what we've done so far. Hopefully bring more for the next meeting. Someone else has some.
Well, it was clear to me at the beginning, there's no way we're gonna defeat the mites, I mean, the beetle itself. That basically, it would be an environmental disaster if we were to get rid of all ambrosia beetles. I'm really impressed that these mites have came in and started dampening the population of the beetle. That's very good news.
It is, and this is a phenomenon that with invasive species, usually at the beginning, you have an explosion of the invasive species, but slowly different organisms that are in the environment start to respond and start to control it. So that's kind of the idea that we have in this project, what we want to explore. If these naturally occurring mites can parasitize the beetles and disrupt disease transmission again.
Yeah, Daniel, is there any indication that the fungal symbionts in these alternative ambrosia beetles has now changed? In other words, Lauricula at first was sort of getting the upper hand in their mycangia, but now there's been enough time, just what you're saying, over time, that's sort of being sorted out and it's losing ground inside.
I don't think there's any hard evidence on that, but I do believe that is happening. For one primary reason is if the pathogen lost its primary vector, these associations between the pathogen and beetle are very strong, have co-evolved for many years. So in avocado, if the pathogen lost its primary vector and now relies on these secondary inefficient vectors, it lost its ecological niches, competitive edge. Now it has to compete with everything. So most likely the pathogen itself is now exposed to many things that before, as having a primary vector that is a primary colonizer, the first colonizer of trees like the red ambrosia beetle, an aggressive vector. Losing that also exposes the pathogen to many of these interactions.
Future.
But that's difficult to prove. Yeah.
Thank you.
All right. Well, hello, everyone. My name is Romina Aziz, and I'm the pathologist, one of the pathologists in the group. And today I'm going to give you an overview of the progress that we have made so far. So just to remind you, my lab has 4 main research lines. One is to find short-term solutions, which will be exploring more synthetic conventional fungicides that growers can use today, right? And so that would be short-term solutions. Then we are also doing research on biological control. Daniel wants to kill the beetle, I want to kill the fungus, right? So we are taking different approaches, but at the end, this will have to be a comprehensive integrated management of the disease complex. And those will be kind of midterm solutions, more sustainable, more ecologically friendly, and hopefully less cost prohibitive. We're also doing germplasm screening, and that will be long-term solutions, right? Very difficult to find another HAS, talking about, you know, overall world production HAS is number one. So very difficult to find another HAS out there that may have tolerance to the disease, but we can look into rootstock, different genotypes of rootstocks and maybe interstocks and so on. So we are doing work on that to find long-term solutions, not only for Florida, but also to for other industries abroad. And of course, we still need to optimize diagnostics and surveillance. This goes hand to hand to the Karen's presentation on offering resources and tools to growers in areas where the disease is not present yet. And so I showed this slide in the last meeting. We haven't found the solution yet, but we have made progress in many fronts. And so if you want to read these papers, you can let me know and I can share those papers with you. There is information on different chemicals that we have tested, the progress that we have done in germplasm screening. We are partnering with collections in Mexico, in Hawaii, in California, to screen these genotypes and hopefully find one that can be integrated into a management that includes genetic tolerance. Very difficult to find resistance, but maybe tolerance will help this, you know, allowing the extended production of maybe infected trees. And so you can just let me know and I can share those papers with you. And so talking about this particular grant, my lab is in charge of Objective 6. We do collaborate with other objectives, as it was mentioned before, but this is our kind of the one that we are leading. And so we are looking into other new chemistries, again, conventional synthetics, but also natural products. And that goes hand to hand with application modes. And so we don't only want to show that a product works, but we need to test if it's feasible to be implemented in our conditions, right? So we need to know if it is worth to do injections, to do soil drenches, and so on. So those two sub-objectives go hand to hand. And then we're also working on biological control, as I mentioned before, looking for other strains of trichoderma that can be good partners in this integrated management. And also we're going to look into peptides, which seems to be the trend in many diseases, right? And so just to remind you, we have revisited a lot of different active ingredients. Some of them were already looked at, but maybe not, but maybe only in vitro. And so we have also conducted greenhouse studies. And so we also expanded this survey of active ingredients, looking for something that not only works in petri dishes, but it can move inside the plant. With the caveat that we are using young trees. And I completely agree that it's really not a natural system, but it's the first step to move forward. And so we did a lot of work and we found one that was flutriafol, which is in the same group as propiconazole, which many of you already know. But we found that it could be absorbed by the roots of these potted trees 50 times better. So we were over the moon, right? Very excited, very hopeful that it will work. We partnered with the industry, so we convinced FMC, which are the only ones that produce chemicals or formulations with flutriafol in the US. So we partnered with them, we partnered with Rainbow, who does a company that works in the landscape, but they also inject trees and they have a formulation for injectables. IFAS supported us in this endeavor. And so we were super excited. I had a new postdoc join my lab, Andrew, who is next to Daniel in the back. And so he's leading this experiment. Anyway, so there are multiple reasons why we believe in flutrefort. One of them that is already being used in nut and fruit trees. So that means that, you know, it's kind of safe, but we still have to prove it that it's safe in avocado. Anyways, very exciting. We went to the field and soil inject and trunk injected the trees, mature producing trees. So that's a better reflection of how things are in nature. And unfortunately, very disappointing results. I know we can't win. And so, yeah, the concentration of the product in the sapwood is extremely low, below what we need for this active ingredient, which is 10 ppm. We need 10 ppm for the fungus to die. We need 1 to hold it in place, right? To inhibit 100% of the growth. But we need 10 ppm to kill it. Well, most of the treatments didn't make the cut. And so we are still sampling because these samples are very expensive to process. So, you know, we send a few first, and then we send more to see how you know, not to invest in the whole set at the beginning. But in any case, the concentration is very low. And so the product is not moving. It's not moving. All right, so now we're going to talk about the biological control aspect of this grant and previous studies. So we just published these studies open access, so you can scan the QR or ask me for the paper. So we did find several trichoderma, which is a good fungus that can inhibit the growth of the pathogen and can become endophytic. Some of these trichoderma are present in commercial products, which is great. But some of them are were also isolated from avocado trees that seem to be holding and not getting sick. Most of these trichoderma come from Carlos de la Torre growth. As you know, he has this integrated management approach. And so anyway, so we show that there are trichoderma that can inhibit 100% of the growth of the fungus. It cannot kill it, but it can inhibit. It can become endophytic, which means that it can colonize internally the plant, but this colonization is very ephemeral. So after 3 months, you don't see it anymore.
So far, you showed that it kills it on Petri plates, right? You haven't.
Correct. That's correct. And so again, the disappointing part is that it did not stay in the plant for many months. So you will have to be reapplying the trichoderma to make it work. But even there, we are not there yet to say the trichoderma is inside and is killing the fungus, the pathogen inside the tree. We don't know that. And so just to keep working on this, I think it's very valuable that we found these trichoderma that can inhibit more than 90% of the growth of the fungus. And if you increase the metabolite, the filtrate in the Petri dishes, it will equal propiconazole. So 100% of inhibition. No fungicidal, so it doesn't kill the fungus, but it inhibits it quite strongly. And so we want to keep working on this set of trichoderma. Now we are narrowing down the species that are able to produce these filtrate, these metabolites that inhibit the growth of the pathogen. And so, Andrew, yes.
A question on the field work you're doing on these are all on established trees. They have been infected, who knows for how long, versus my idea, when you have newly planted trees, potentially, maybe that aren't infected. I'm wondering if some of the lack of effect in the fungal removing the killers is because they're infected. The tree has already had its allergic reaction shut down and doesn't let stuff through as well.
So that's a good point. I don't think that talking about the chemical movement, right? And you're right, when they get Laurel Wilt, those tyloses will prevent movement in the vessel. So it doesn't matter if you put plutonium, it's not gonna move.
Yes,
Right,
do we
because
have an
it's blood.
opportunity with maybe young, newly established
Yeah,
girls
so
to do these?
That's a good point. So the young potted plants have a different vasculature,
Yep.
and it's like more like a, like a straw, not like a palm, but it's like more movement. These older trees in the field, and I'm gonna talk a little bit about the microbiome that we are working on. There is a lot of wood decaying fungi, which cause localized compartmentalization of the vascular system. So maybe these trees, as you are mentioning, have been through a lot, right? Because there's a lot of fungal decay everywhere. And so the vascular system might not be as a freeway as a young potted plant, right?
But I'm talking now about essentially young planting
Yeah.
in the field.
Yeah, that's a good.
We can find that
Yeah.
somehow, get that going.
That would be nice. We could do we could apply the treatment and then see if the movement is better than an adult.
And
it
effectively as
yeah,
it regrows,
yeah,
you can
that's
see
true.
maybe there's
Yeah.
less resistance
Yeah.
to the.
Well,
Yeah.
when we measure
That's a good point.
transpiration of mature trees, it's often even higher than potted trees, they're still transpiring wide.
Yeah. Yeah, but I wonder if, because we found a lot of wood decay inside trees, and I wonder if that movement is being challenged by
It would
these
depend on,
conditions.
where you inoculate them, what silent vessels.
Yeah, I mean, we're doing the active silent, yeah. So that should be new silent. It should be young and anyways, but there is a lot of difficulties.
Young and innocent and so.
Yeah, hasn't been.
It stinks about it.
Yeah. Anyway, so because we were excited about this metabolite production of this trichoderma that have been isolated from the inside of avocado mature trees in the field, you want something that can live inside the trees, right? So continuously be pumping these antifungal metabolites. And so we expanded the search and we got a lot of isolates from collections, and we screened again, and actually the ones that we had previously are the best. So we're gonna stop looking for more and then concentrate it in the ones that we have, which we know that they can tolerate the environment of South Florida. And so we are moving into kind of optimizing the production of metabolites. So this trichoderma, which is the good fungus, is producing these metabolites, right? And what we want is to see if we can make these metabolites stronger or maybe different to the point that they will kill Harringtonia loricola. Because now the fungus is not being killed. It's just being, suppressed. And so we are going to try 3 different experiments, co-inoculation with the pathogen, to see if the pathogen elicits a different production of metabolites, a stronger production of metabolites. And so this is being done in vitro and in progress. And so my thinking, so we have 3 methods. One is cultivating with the pathogen alive. The other one is deactivating the pathogen, and maybe the chitin and the cell parts is going to induce more production of the metabolite for trichoderma. And then maybe Harringtonia loricola also produces some chemicals that are sensed by trichoderma, in the hope that we will find something that is fungicidal. My thinking is that delivery is very difficult. So I think that if we find which metabolite is inhibiting the growth, we can synthesize it and then just apply it as it was any other chemical, right? And so we'll see. That is in progress. Another thing that we're looking at, another question, is the microbiome. And so we are doing this in collaboration with USDA ARS. And so we now have a lot of data on what organisms are living within avocado trees, mature trees, that are diseased or no disease. So do they have Harringtonia loricola or they don't? So to see if there's any differences on these two treatments or two scenarios and see if there's something in the microbiome that is making the tree, I think, less appealing to the beetles, or perhaps holding the pathogen from really multiplying and moving freely. So, long shot, but it's definitely going to give us an insights on what's living within. So far, we have found differences in between trees, the stem, leaves don't differ, roots don't differ, but the stem of trees, the composition of the microbiome is different when you compare trees that are infected versus trees that are healthy, which makes sense, right? If you put a pathogen, everything gets unbalanced. But we want to know which are these different. So we have to dig into this data. Finally, we are going to continue to look for other organisms that can inhibit the growth of the pathogen, including bacteria. And I presented this last time. In the research that we did with certain entomopathogenic nematode-associated bacteria, we did not find any inhibition for Harringtonia loricola, even though these compounds do inhibit other ambrosia fungi. So Harringtonia loricola is pretty tough, but we are going to expand this search and already have collaborations in place to do so. And we are also going to look at peptides. Again, this is a novel approach in Laurel Wilt, but it's been very popular in the studies that target other diseases. So I'm recruiting a student, and she's going to join the team very soon. And she's going to be working on these first in vitro, then greenhouse, and hopefully in the field. So I really have high hopes in testing these peptides, many of which have trichoderma origin. And so with that, thank you to all the grants and all your support. And I'll take questions if you guys have. I want to mention something. I really I'm reflecting on your comments, and you said that you agree that the pathogen is present in these trees that have been stamped, right? And there we agree. We are in agreement.
Right.
I went to visit the plot from Brooks that we have inoculated, and they had been injected in the past, and they got sick, and they were stamped, and they were regrafted, correct? Yes, and actually they are looking, for the most part, good. For the most part, there were some that did not recover. So I think that the strategies of the fungicide that has, so how do I say that? So none of the trees were protected by the fungicide applications. However, the symptom development was really suppressed, which means that the population of the fungus has been suppressed, which means that it gives the tree the chance to build more xylem. And as you mentioned, when you stump, you have the root system that allows, and because it doesn't have to invest in canopy, it invests in trunk, right? Layers. And so I think that there is something to it. I think that all these strategies layered, plus the grafting, which is very localized in a section of the vasculature, will perhaps give the chance of the tree to keep living. Now, my concern as a pathologist is to leave the infected root system in the orchard. What is the risk of leaving a stump and root system infected with the pathogen versus removing the entire tree and plant a new one?
Okay, can I answer that?
Yeah, of course.
Number one, economically. Number one, it costs about $100 a tree to remove a tree and dispose of it, okay? Number two, we never injected any fungicide in our trees. And I showed Brooks from the beginning. I showed Danielle and Jonathan that they were regenerating. And I will admit that not all of them made it.
Yeah, okay.
But
okay,
if
we
they
have
can make it,
understanding.
okay,
Yeah,
listen, if they can make it another 10 years,
uh-huh,
that's 10 years of production.
Yeah.
Yeah.
That's an economic plus. Okay, so they may die in 10 years. But economically,
Yeah.
the slash and burn is a
Yeah.
very
no, I
bad
agree
thing.
with you when you look at the tree individually. But now I'm wondering, and maybe the economic team can look into these scenarios, right? What about the adjacent trees?
Grafting is the thing
Yeah,
that
and listen,
worries me the most.
Exactly,
And then
exactly.
people
So
go
we know how to control
crazy
it.
trying to cut the roots, okay? It didn't do any help. It didn't help at all. It's obvious. Sick, older trees are more susceptible. Regenerated trees are less susceptible, and young trees are less susceptible. But they all gonna get it.
Yeah.
One good thing is the beetle population is reduced. But no, the root grafting is BS as far as I'm concerned, because I've seen too many people cut the hell out of the grove and it didn't do them any good, like Murray Bass, right across the street from Trek.
Yeah, but I'd say, Mark, so he did the trenching, so we have this idea, if we sever the roots between the.
Yeah, well,
trees, they
did
won't
it do
move.
them any good?
Okay, the only thing is, he trenched all the way around the trees, but when it came to where the irrigation system was underground, he didn't cut that.
Which the roots like that.
Well, they follow any.
Oh, they love
old-
it, right?
Yeah.
I'm just saying.
In any case, I'm saying that I think it's a.
Economics is the main thing here. Basically, the industry has almost been killed by the slash and burn. We have had to, in industry-wise, figure out a way to continue in production. And if we don't produce, and all we do is spend, we're dead.
Yeah, so,
You mentioned something that stuck with me about the cutting and then regrafting the tree. Yes, they're just isolated sections.
mhm.
So have you tried maybe putting more grafts than you, normally how many grafts would you put in a stump?
Three.
On a
I
stump?
think we have more.
What if you put 10 or 12?
Yeah, exactly.
And then
So,
observe it as soon as you see a
Yeah.
symptom, get rid
So-
of that one, and then maybe you'll have health.
so let me,
I
so
don't
when
know.
we did the
Yeah.
experiment at Trek, which we drench, and then all the trees got sick, the 70 trees got sick. We stumped them and we let them resprout. More than 80% came down, but we let it resprout. Now, what Brooks is doing
No, they're
is
different.
grafting,
Yes,
which narrows down the
Mhm.
vasculature.
yes.
And the chances that you're gonna get a layer that is
Yeah.
That's
healthy.
infected. Yeah, exactly.
Yeah. Now we show also that the pathogen can move from infected layer into new layer, but it's a chance.
Mhm.
It's a game of chance, right? So my questioning is, what's the risk economically of going that route?
I would say adding more scions in there
maybe.
is not that much of a loss
But I would
Production,
just say
for maybe a
too,
gain.
we also found the vascular system of sprouts coming out of a stump is like a young
Oh, yeah.
tree,
Yes.
grafted tree. It's different. And that xylem regenerates very, very quickly.
Yeah.
Right?
So
So
you're
that's,
saying the growth is
it's out-
outpacing the.
yeah, it's
advancing.
outpacing the events. It's when there's some transition that happens that stops that proliferation of xylem development, that the fungus can catch
Yeah.
up, and then.
Yeah.
That's
Once
why
it
you see
becomes
the limbs that are
woody,
6 feet tall after 2
And then
years,
suddenly got
all of a sudden they go down.
And so I don't know how many production seasons are until it gets to that diameter or that maturity. And so, again, I am telling you what we have done, what it protects or not from the disease, it does not protect. But when you stump, the tree has less resources to put in the canopy, so it will put it in making itself thicker.
So our next speaker is Jeff Rollins, and you're connected, Jeff, everything's good? That's
Okay.
his speaker.
Can you hear me okay?
Yes.
All right.
We'll
Yeah,
let you-
yeah. Thanks, Jonathan. Yeah, I appreciate the opportunity to share research we've been doing in my program. Sorry I couldn't make it in person, participate in that dynamic discussion. But kind of building on what Romina was saying about long-term, midterm, short-term kinds of solutions and insights. So my research is really focused on long-term solutions, and that's to investigate mechanisms of host resistance and susceptibility, and also to use a model host species. In this case, it's Nicotiana benthamiana, to test the endogenous and exogenous RNAi strategies for developing disease resistance. So I'll give you a little bit of update on the work we've been doing under these two objectives. The first, investigating these mechanisms of host resistance and susceptibility. So we're looking to compare the dynamic colonization, the Tyloses development, and gene expression in North American susceptible, meaning avocado and swamp bay trees, versus southeastern Asian resistant, and this is primarily camphor trees. And so we're looking at trying to compare and contrast the differences between this resistant Southeast Asian species and these susceptible North American species. So the hypothesis is that the pathogen colonization and the associated host defense pathway induction are similar in both susceptible and resistant, but we think the timing and the magnitude perhaps are what differ between these two. And what we hope with the outcomes is that an understanding of these host responses related to disease resistance relative to susceptibility will provide us with tools for more efficiently screening avocado cultivars for resistance traits. If we can understand what the difference is between what a resistant reaction looks like and susceptible reaction looks like, we could look for that to see if we have any cultivars or any other germplasm that may show these more resistant types of reactions. So the specific objective 4A is comparing these dynamics of colonizations and Tyloses development and gene expression. So work we've done here is we've done inoculations of the avocado, the swamp bay, and camphor trees. I have 2 graduate students that started in January of this year, and they've done the RNA extractions, the purifications at the 24-hour time point. So we're going to look very early in the interaction between the fungus and the host. We've done the RNA-seq. We've gotten back our reads for that. We got really good read, both the depth of read, like 71 million on average with 3 replications. For the camphor inoculated with Harringtonia loricula, 67 million on average with the water controls. And then with the Persea americana, the avocado, we got an average 70 million and 90 million in the controls, and very high quality RNA. So we're in the process now of mapping these reads, these RNA-seq reads, and doing the differential gene expression analysis. We're also looking at the colonization dynamics between the resistant and susceptible, so avocado versus camphor trees. And so we take advantage of our GFP labeled pathogen to basically look over time. So with different time points, we dissect the trees and we plate them out and look for GFP expression. So the accumulation of the pathogen growing out of these stem segments. And we do that both here in a macroscopic level, just with a UV light, but we also look under the microscope to see the individual hyphae growing out for confirmation. And then we're beginning to look more at the cytological aspects of the interaction, again, between resistant and susceptible. So we're looking at Tyloses, reactive oxygen species accumulation, lignin deposition, et cetera. So we're, again, looking at these and just trying to understand, is there differences in the mechanisms, or is it differences in timing between resistance and susceptible? The second objective is objective 5, and this has 2 components, but it all revolves around using this model system. So we use a non-tree system so that we can make faster progress in understanding disease resistance. And some of this work came out of previously funded work identifying targets within the pathogen that are vulnerable for us to exploit. So things that are absolutely required for the pathogen to colonize the tree to cause symptom development. So we've identified one of these targets, and we're looking at other targets as well, but this is a proof of concept here. So we're utilizing the model host species, and we're going to be testing both endogenous, so expression by the plant, and exogenous, meaning us applying or delivering small RNAs and double-stranded RNAs, and so that we can develop disease resistance. And so for this objective 5A, we're comparing external. We kind of want to just make sure our system is really a good surrogate system for studying Laurel wilt. So we're looking at comparing to Benthamiana, what we know about the colonization by the pathogen and external internal symptom development, comparing that to susceptible Lauraceae trees. So we want to make sure the dynamics of colonization and our model susceptible host is equivalent to that we see in susceptible Lauraceae trees. And so our hypothesis is that indeed colonization, symptom development, and the pathways underlying susceptibility in our model host are shared with the susceptible Lauraceae. And what we hope the outcomes will this objective will be is that the appropriateness of this Nicotiana benthamiana as a model host for Laurel wilt will be determined. And the use of this non-woody host plant has the potential to speed the rate of discovery, provide insights, and hopefully effective disease management long term. So again, within this, comparing the external and internal symptom manifestation and colonization, we've done these experiments. We've been doing these experiments since last year, and now we've really ramped up with the new graduate students on board. And so we've begun by doing colonization studies within Benthamiana. So Benthamiana grows about 30 centimeters in height. We grow the plant so we get much more of this long stem architecture on them. And we've done the colonization studies and seen that the dynamics of colonization are similar to what we see in potted Lauraceae trees also. And so basically the green here shows where we recover the pathogen and replicated inoculated plants over time from 3 days to 28 days. And so we get really extensive colonization of these Benthamiana plants. So we're repeating these experiments as we go. We're also doing titer studies, so we can put as few now repeatedly, and we're repeating these studies also, as few as 300 spores within Benthamiana. So we really want to make sure this isn't just some kind of overreaction of the Benthamiana plants due to giving it just excessive amounts of inoculum. So we've done these titer studies to demonstrate, again, these are water controls. This is our pathogen inoculated with 3,000 spores. You can see the wilting on the leaves and the necrosis of the leaves here. And this is actually inoculated with a non-pathogen, the Harringtonia awakate. And we've done this with varying amounts of spores as well, and it always remains healthy like our water controls. So the other aspect of developing this as a model system is to look at the reaction of the host to the pathogen. And so we're also looking at gene expression here. We're doing an RNA-seq experiment. And again, we have our 24-hour time point that we've completed. And we've got the RNA-seq reads back. And we have very good reads here with 3 biological replications. And we have Nicotiana benthamiana inoculated with Harringtonia loricola. We got about 51 million read pairs back, very high quality, and then a similar number for our non-pathogen interaction, and also in our water controls here. So we're in the process now of mapping these reads and looking at differential expression analyses. The other aspect of this objective 5 is to determine if these exogenously applied or transiently expressed or stably expressed double-stranded RNA molecules can protect plants from lethal Laurel wilt disease. So we're beginning this by, and this is in Nicotiana benthamiana, so in the laboratory, first, the big question we had was, it's been demonstrated in many other fungi, but not in Harringtonia loricola. Can Harringtonia loricola take up double-stranded RNA? So we've done these in vitro studies using labeled double-stranded RNA. And we look for uptake. Here, this is more of a hyphal culture. grown here, and we're using a control Sclerotinia sclerotiorum, another fungus which has been studied fairly extensively for its ability to take up double-stranded RNA and for disease control to be effective by applying double-stranded RNAs either exogenously or those that are produced by the plant. And you can see here the Sclerotinia hyphae take up high amounts of these double-stranded RNAs, and we can detect it also within specific hyphae of Lauricula. So it doesn't take it up as efficiently, but it is taking up double-stranded RNA. And we're now looking at some of the different developmental forms of Harringtonia lauricula. What we primarily see within the plant are the spores, and the spores have to germinate so that they can overcome barriers like perforation plates and move through pit membranes to move from vessel to vessel. So we're looking at conditions that more kind of replicate that type of growth, the type of growth we see in the plant, to see that also double-stranded RNAs are taken up in those forms of the pathogen. So we have one target, and that target is a gene from the fungus that's known as MET-R or CIS3, and it's one that controls sulfur metabolism. So we've made mutants of the pathogen and demonstrated that this gene is absolutely required for the pathogen to cause disease. And so we're looking to down-regulate that in the plant. And so in the laboratory initially is where we're doing these experiments. And so we just synthesizing our double-stranded RNAs. And at this moment, we're using this petiole infiltration technique to deliver the RNAs into the plant stem. And then we've been able to observe these using our fluorescently labeled double-stranded RNA. And indeed, we find that we have high accumulation. This is 24 hours after treatment. We have high accumulation within the xylem vessels, which is where we're going to find the pathogen as well. So the next step with this is that we're going to see what prior treatment with double-stranded RNAs does to infection. So we're going to infect these with Harringtonia lauricula and determine if we get some protective effect. Ultimately, we'd like to, because delivering these is much like delivering a fungicide, maybe even more complicated with their amount of time that they might persist in the environment and persist in the tree, may be lower, much lower than we would see with a fungicide. So ultimately, we would like to get the tree to produce these double-stranded RNAs. And to that end, again, in Benthamiana, we're making constructs to stably express our double-stranded RNA. And this just shows the cloning steps that we've completed at this point in time. Ultimately, we're going to transform our Benthamiana plants and get stable constitutive expression, and we'll challenge these with the pathogen as well to see if we can achieve a level of disease resistance with these plants. So that's kind of the things, that's the main objectives and the progress we've made over the past year or so on this. And I'm very happy to take questions if you have them. Thanks.
Anybody have any questions for Jim?
I didn't understand a word there.
I guess I do. If this is successfully, proven successful, implemented, does that like make avocados then a GMO product?
Yeah, so it seems like I got echo. That's bad. It's gone. Yeah, so that would be a GMO for sure. If we were to do the strategy, to work through the strategy that we're talking about here, it'd be a GMO. One of the questions would be regulatory wise, and if we were to able to engineer rootstocks and not scions here, how that might change the regulatory landscape. So that's one thing that we're thinking about to that notion. But I think as we've seen with citrus greening, acceptability of transgenics, the idea that transgenics may become part of the our fresh fruit products, et cetera, seems to be gaining traction in light of the economic concerns of being able to grow citrus, and hopefully avocado won't be there, but that's, yeah. Long term, that's what we're thinking, is a transgenic approach may be the approach that we may need to really have effective management of the disease.
Anybody else have any questions? Sure. Thank you.
Yep, appreciate it.
So Tom's gonna talk a little bit about status of some of the applied research, and you've heard from the researchers mostly beforehand, so I don't have to go into real detail. But I want to acknowledge all these groups and people for their support for this project, and the participants, the researchers, ag manager, FDACS, the administrative committee, and also the advisory council, advisory panel members. Okay, so Romina already talked about her testing, so applied research participation. We collaborated with Romina's lab, new fungicides, formulations, germplasm screening. I was gonna mention one of the ones that you saw that showed some tolerance to Laurel Wilt is called Colin B33, which was from the USDA. We have the germplasm in the US. And then also Daniel participating with him with this groves, with different pruning practices and management practices, looking at, is there things that we can do in the grove, cultural practices that are, now skirting is not common here. For skirting the avocado trees is not a common practice. It is in other fruit crops, more temperate fruit crops, but this may be something, so this is something that we're looking at to see, does that also enhance the depression of beetle activity and beetle movement? So that's one of the things. So we're working with Daniel's lab, or Daniel's leading this. And also, I just wanted to mention, so we have this cyan rootstock field trial here at Trek, which was put in with the last grant, which the last grant was really not only worked on Laurel Wilt, but also worked on Phytophthora root rot, which is another fungal pathogen of avocado trees, and is especially detrimental if we have flooding events or saturated soil events, or if the water management district holds the water table up too high for too long. So we have a rootstock cyan study using germplasm from rootstocks that were developed at the University of California for tolerance to Phytophthora root rot. So we have this small planting. It was put in August, well, basically May to August 2022. And these are the cyans over on the left: Donnie, Jem, Haas, and Reed. And then there are 6 rootstocks, 5 of which are clonal from the University of California breeding program, and then our Walden seedlings. So there's a lot of combinations. And this is a lot of and some of the material Bruce's work for his rootstock. And so I was just, okay, so we've been taking some fruit counts, and then I converted this into pounds of fruit per acre by this cyan rootstock combination. On the far left is the Donnie on the different rootstocks, and it's year 2024, the number of fruit, or pounds of fruit, sorry, and then orange is 2025. So you have Donnie first, and then Jem second, and then Haas is third, and fourth is Reed so far. This is just production, these very 2 early years. This is not anything definitive, but just to show you. The most productive so far has been Reed, then Donnie, Jem, and Haas. The story of Haas in South Florida, I'm talking about deep South Florida, Homestead area, has been it's never really done well. There's a reason we don't grow Haas, partly because it's not adapted to our conditions here. We do see Haas being planted in Central Florida, so there may be a different answer when we move north. However, and you can see Donnie and Jem were most productive on particular rootstocks, and Reed so far was most productive on this one of the clonal rootstocks, the number 40. The other thing that we've noticed is this tree death and limb and shoot dieback. So we had, out of the 48 Haas trees that we planted, 2 have died, and it doesn't appear to be rootstock related. Romina's looked at it, and we've looked at it numerous times. And the percentage of trees with dieback out in that planting, anytime you want to visit it, just let us know, has been none for Donnie, Jem, and Reed. But 60% of the Haas trees look like that tree on the left. They have a limb all of a sudden fires up and dies. The rest of the tree seems okay. this is some early symptoms on the right-hand side showing these early wilting symptoms, and that branch or limb is going to die. You'll see this dieback throughout the canopy. The other thing we've noticed is we were out looking at the leaves, of course, we're out looking at the trees, and it seemed like this leaf distortion, we see these distorted leaves, and we're like, okay, so is this herbicide damage? What is it? Is this viral? You know, what's going on? So we took leaf samples to compare. We did this on Haas and Gem. So we compared leaves on the same trees that showed symptoms and leaves that did not show symptoms. And so then we compared the nutrient analysis between these. And the NPK was normal within published ranges for the percent nitrogen, phosphorus, and potassium. Our calcium readings on all the trees were low on the low side, but within normal ranges again. Boron seemed to be slightly below normal ranges for all, whether they were symptomatic leaves or not symptomatic leaves. And then, but all trees, again, all trees were deficient in manganese and very deficient in zinc and copper, which is, I didn't expect at all. So a little bit of a mystery, not sure why. I've not really seen, and you guys would know better than me. This does not look like typical zinc deficiency symptoms to me, or manganese at all. So I'm not sure really what's going on. So what I am doing though is we are increasing our minor element applications to see if this doesn't make a difference. So we'll sample in the future to see, does this correct it and correct these symptoms? But it's just something, an observation to let you know what's going on out there. The extension component, so the website has been partially updated. We have ongoing updates, looking at the literature, the videos, and information. Fact sheet recommendations are going to be in revision. We have already planned future workshops and seminars, and you'll be hearing more about those, of course. And we will be notifying people through email and recommendations as needed.